My name is Michael Maguire. I'm CEO of Avectas. I'm delighted to be presenting at the Cell meeting on the MESA and looking forward to updating you on progress in the development of our cell engineering platform, Solupore. Solupore is being developed to enable the next wave of cell therapies. We've seen the commercialization of the first generation of gene-modified cell products. These are typically single transgene modified T cells addressing blood cancers. They're typically autologous products. It's our view that the next generation of products will need to address a greater amount of the tumor burden, so to address solid mass tumors, and also to move from an autologous manufacturing process to be allogeneic in nature. So those two drivers or needs are creating the need for more complex engineering. And more complex engineering means more editing. It means the coexistence of viral and non-viral processes or delivery within one process. And all the time, the viability of the cells needs to be maintained such that when the cells are infused into the patient, they're as functional as they possibly can be. So engineering is getting more complex, but the cells which come from that engineering process need to be minimally perturbed and need to be as functional as they can be going into the patient. And Solupore, our platform, is developed precisely to meet this need set around complex engineering. So a word about the business. Avectas is based outside of Dublin in Ireland. We're at J Labs in Toronto, and our chief business officer is based in Boston. We've 35 employees. We've raised over $40 million. We've a very experienced leadership team and board from the drug delivery industry, and we've a world-leading scientific advisory board. Our business model is to partner with therapeutic developers or high-tech or advanced CDMOs who are building out capability in advanced manufacturing. So I want to show the first embodiment of the Solupore platform. This is the research tool. So the Solupore research tool is a gateway instrument. This is the second generation of research tool. During the course of 2020 and 2021, we placed 10 of the first generation of these devices in labs globally. We garnered the feedback from the use of those devices and inculcated that into a second generation. So this is a brand new device. It's very tech transferable. Very easy to use based on learnings and primary user feedback. We've developed a suite of technical support and SOPs and data and so forth. And this is the device which will underpin research collaborations. The initial part of commercialization is, of course, the feasibility study and being able to place an instrument or technology in the hands of the partner so they can build data under their own roof, if you like, is critical for us. So that's the role of the research tool. We then have the Solupore SUS, the clinical system. So this is the closed GMP aligned system. It's a clinical grade engineering system. It's automated, it's highly reproducible. It's designed precisely to affect the complex editing that I've spoken about in my introduction. It can deliver a wide range of cargoes to cells, and indeed it can deal with a wide range of cells too. We can deal with messenger RNA, RNPs, including Cas9, CRISPR-Cas9, and also deliver plasmids and other molecules. So this is the centerpiece, if you like, of our clinical offering, and it's on this device that products will be manufactured, non-virally modified and engineered before they're moved on to the next part of the process, be that proliferation, fill freeze, or whatever that is. So a brief word on the Solupore process. Cells are introduced into the chamber on the left-hand side. The cells are in suspension, and when they arrive in the chamber, they're immediately taken out of suspension onto a highly engineered filter. They're then targeted with an atomized or dropletized spray of membrane permeabilization solution, which also includes the cargo to be delivered. Literally on contact, the cell membrane is transiently permeabilized at the same time as the cargo is concentrated at the membrane. The means of transport is passive diffusion over the membrane. And when this process, which is very short, is finished, the cells are immediately resuspended. The process end-to-end is 7 minutes. That's presented here. Most of that time is actually getting cells into the device and out of the device. The actual transfection step is very short. Critically, this process is really well tolerated by the cells. We enjoy high transfection efficiencies, high viability of cells, and the cells are highly functional coming off this process due to the gentle nature of the process. The Solupore system has 3 key elements, I guess, and those are the delivery solution, the membrane permeabilization reagent. There's a single-use assembly, which needs to be sterile and so forth. There's a multi-use controller as well. We've a well-evolved regulatory platform, and delighted to update this group that we met with the FDA in March 2021. And we're now continuing on our regulatory pathway, suitably guided by that meeting and other input that we've had in the period since. We'll file DMFs and also have the suite of documentation available to people in different regions working perhaps outside of the DMF system who nonetheless want to file INDs. So we can span the full pathway from a product concept through to commercialization of a product. A word on the scientific performance. We show on this slide and the next, actually, focusing in specifically on complex engineering. So just taking a little bit of the data which we have to hand out to, on the first left-hand side, showing multiplex delivery of CAR and GFP mRNA, once again seeing high viability and very nice expression in the double positive phenotype on the in the graph. On the right-hand side of the slide, we see sequential delivery. So we're doing a track edit and then an mRNA, CAR mRNA delivery, once again seeing very nice viability along with good expression. Once again, on this second slide, we see that high viability is maintained at all times. On the left-hand side of the slide, we see a range of different targets edited in T cells and one in NK cells. These edits are with partner companies, so we've anonymized what the targets are, but the edits are, efficiencies are presented there. On the right-hand side, we show moving beyond CRISPR to different editing platforms, some fairly generic, some proprietary, and we see nice editing efficiency as well. We've produced a large body of data, which we published in Cytotherapy in April in a paper, and you can delve deeper into our data set beyond this particular update to the MESA meeting. So in the spirit of an update, what are we working on right now? So we're developing a very exciting data set at the moment. We're focused on hard to edit cells, so cells that are can be finicky, difficult to handle. We're looking at multi-edits. We're looking at sequential edits, one edit after another, and seeing how we can build on that. We're looking at measures of gentleness, both the function of the cells coming off the system and other measures of gentleness in the process. And we're doing extensive process development on the SUS system, which I've shown, and also on the research tool. I also thought that it would be valuable to update on our progress towards GMP alignment, and maybe just to put up some of the themes which are critical here. You know, in the sense of hardware, we're working on documentation, obviously our safety testing, and the suitability of the design. So reviewing the design for clean room use. So all sorts of, does this device produce particles? Are there, is it cleanable? All of these very practical things, but its usability in a class C or D background is critical. So that type of work has been done at this stage. On software, our Chapter 21, Part 11, compliance, tiered access to the system, and the documentation of that software is critical as well. In terms of regulatory pathway, our folio of documents is evolving, and we are working with a regulatory advisory in terms of constructing those DMFs. In terms of consumables, some of the considerations, obviously our system closure, sterility, and particle-free status of the consumables. None of these things are trivial. They have to be validated and presented in our documentation suite. And then lastly, in terms of reagents, formulation compatibility. So as we put different cargoes in, how does our membrane permeabilization chemistry work with those? The stability of our delivery solution. So practical things like, you know, what's the shelf life? And what's the container presentation going on to the device. So all of these considerations were well progressed in, but I thought that might be useful at this meeting to present how we're thinking about our system. We know that there are several other systems under development out there, and that this might be a useful ready reckoner to present what's important from our perspective. We continue to work with the key CDMOs and partners in terms of augmenting our efforts to research, and we continue to invest in our IP portfolio, which is growing. We have an excellent scientific advisory board and management team, and there'll be many familiar faces on our scientific advisory board. We also have a technical advisory board, and between them, we're evolving the tech, drawing on the best advice available to us. So in summary, Solupore is it's a new automated cell engineering platform. It's being developed to manufacture the next generation of cell therapies. It's extremely well tolerated by cells, and we're showing increased efficacy and higher proliferation of cells post-processing. For autologous therapies, we can deliver to fragile cells, for instance, T cells, some T cells, subpopulations, and NK cells. And we're also concerned with monitoring those cells out after the process to see how functional they are. We can affect multiplexed or sequential edits for allogeneic therapies to potentially address solid mass tumors or in tough to edit cells. We can deliver a variety of cargoes individually or in sequence, and we have a series of anticipated regulatory filings coming up in 2022. I'd be delighted to speak with anybody who's developing a therapy or who's interested in a next generation non-viral or viral and non-viral platform to affect complex cell engineering. Thank you very much indeed. Enjoy the rest of the meeting.